And today we're going to kind of slow down and look at a piece of biotechnology. This is not specifically a living thing. This is how we use DNA to help us with our real world problems, supposedly, of the century. So let's just read and figure out. So you're going to notice again, just like yesterday, we've got these goals they're not our purple goals, right? So that means today we're moving up that triangle. We're going to take some of those things that we've learned in the past couple weeks, go and take it up and try to have a greater understanding of and process and just chew on some of those things. Luna, will you read our goals for us today?
Hopefully.
I hear you now.
Let's try this again. In what direction do DNA pieces move when electricity is applied to them? I cannot see that question, but how is gel
Electrophoresis.
electrophoresis? Phoresis. Okay, phoresis. Rhesus pieces. Used to help find someone guilty or not guilty of a crime.
Awesome. Way to work. Those are some tough words. Yeah, so everybody say gel electrophoresis. She's like Reese Pieces, Rhesus. Yes, you're right. Electrophoresis. This is a lesson that I haven't typically spent a ton of time on in the few past, but we want to do this. I want to tell you the last 2 or 3 times that I've proctored the tests for the OST, there are electrophoresis questions. Okay, so let's just understand this. It is a technology that we use to identify somebody, and we touched on that yesterday. And I just want to take a little bit more time and dissect it a little farther so that you feel confident. Ooh, a lie detector. I wish it was a lie detector test. No, I don't wish. A lie detector test is another tool that they use, but this is a little different. So, a lie detector would help us if we had someone that we thought was a suspect for the crime. All right. In this case, we don't have a suspect yet. All right. What we're going to use is a fingerprint. It's been a while ago, but when I was in high school, someone broke into my car. All right. They broke into, yeah, let's just maybe halt the doodling for now. It's just so we don't distract people. Okay. Nothing's inappropriate or whatever. It's totally fine. Let's just maybe not so we don't distract people. So I was in high school and someone broke into my car. So we had to call the police. The police came out and they took some adhesive paper and they were sticking it to the surfaces of my car where they could find some fingerprints. All right. So fingerprints are used for a variety of different reasons. All right. Oftentimes we have fingerprints on file. So fingerprints can match up with other known fingerprints, right, to give us DNA or to give us identity confirmation. Another way is that there is DNA in our fingerprints. Sometimes is DNA left behind. So when they use that adhesion, not only do they get the copy of the imprint like you see there on your screen, but you can also get actual fragments or pieces of DNA or cells, right? So what they do is they'll take those cells, and this I would say is a little bit bigger than like the size of a paper cutter. Have you seen those big paper cutters with the handle? All right, we're talking about a bigger machine. And this is actually like a small tank. All right. Now it's a thin tank. We're not talking deep water. All right. But what we do is we put the DNA sample into wells. These are wells down here, these little rectangles that you see. All right, so we discard the DNA into the wells. And then when you plug them into the power supply, the electricity moves from this side to the other, from one side to the other. The electricity is moving through the water. All right. The reason the electricity moves through the water is that that's the way of pulling apart pieces of DNA. All right. So what we'll do is put the DNA in a well, run the electricity through, and the DNA will separate. The heaviest will be closest to the well, and the lighter sections will fragment out to the top over up here. All right. So it'll give us a look at the DNA fragments of the suspect, right? We have a suspect because there is a fingerprint on my car that contains DNA. So now we have the electrophoresis to pull it apart, and that helps us match up the DNA. Sometimes these are people that are like repeat offenders, so we have copies of that on file. Maybe there's multiple fingerprints on the car, so they take 2 or 3 like samples of that because maybe it wasn't just one person, right? Of a house well. That's not too bad though. Like a house well is kind of similar, right? Because it's a deep place, a deep hole where you put something in it and water's in it. In this case, it is DNA. All right. So as that fragments out, it looks like, let me find a picture of it. Okay, here it is. It will look like see, this is the actual tank. You can see the wells right here. All right. You actually pipette into that. This is a little pipette. It's like a little needle that we use in the laboratory, right? And you put that in the well, right? Carefully place the pipeout tip over the well and expel the sample. All right. The electrophoresis buffer it's like a water gel, right? Because electricity travels through water, we're able to see that. Let's pop this up. All right, this is the machine. All right, these are the electrodes that attach to the machine to send the current from one side to the other. And then this is what the DNA looks like as it separates. Okay, so what this is, and let's make sure in chat, I'm trying to explain this to you, so I feel like we might be distracted in chat. All right, this is what it looks like at the end. All right, you'll have a marker that kind of gives you data points, okay, where things should be or are, and then these will be what the samples are. All right, you'll get different pieces of DNA that fragment out from there. Now, yesterday we looked at a sample. Let me see if I can find it that looks similar, maybe like this. All right. These are the suspects. This is the blood stain. But what you're looking for is similarities, right? You're going to look for things that are similar. If you have a suspect and you have like a blood sample, right? You can start looking for DNA that matches up with those. Or if it's something we have in file. So what I want you to do is I want you to look at this blood. This is blood that was found on the scene. All right. And these are fingerprints of from DNA or DNA from fingerprints of suspects. So what I want you to do is look at this pattern, this banding. Yeah, somebody's on it. I want you to look for the same banding in the suspects. Look at 1, 2, 3, 4, 5, 6, 7. Who has a similar banding pattern as the blood stain? Yeah, I see lots of you saying 3. So look at this. See how the blood stain.
I don't know. I don't know what my blood stain is. What I have is a little white and a little bit of red.
Well, you're right. So Jonathan, what in this case there would have been a crime scene where there was blood on the scene. So like if somebody was hurt, right? And maybe there's an instance where there was a weapon left or there was a drop of blood on the floor, right? What the detectives will do is they'll gather that sample with gloves, with tools, right? And the same thing with the fingerprints. They'll get those, and then that they'll bring them to a machine like this electrophoresis that we just looked at, this gel electrophoresis, and they'll run them through that. So it's going to take the DNA that's in the blood and the DNA, Mila, that is in the fingerprint sample, and it's going to look for a match, right? So in this case, this was blood from the crime scene. Number 3 was a fingerprint from the crime scene. When we put those samples into the gel electrophoresis and run it, we see that the blood stain DNA and the fingerprint DNA match up. Right? This goes all with what we've been talking about, genes and gene expression. We talked about how like our eyeballs and our intestine and stomach cells all have the same DNA in them, right? It just turns some of it off and some of it on. So in this case, this is saying the DNA from the blood and the DNA from the finger, they have the same DNA, right? So when you run them through the electrophoresis, that is what will give us the match. Yeah. So we would say on a quiz question like this that the DNA matches suspect 3. I tell you, almost every year I see something with the gel electrophoresis on it. So I want you to know the gel electrophoresis has to do with what? What is it looking at really closely? A 3-letter word. Yes, Jenna. DNA, right? Gel electrophoresis is looking at our DNA and our DNA segments. And we can match those up, right? Because all of the cells in all of our body and all of our body fluids have the same DNA in them. So DNA stands for deoxyribose nucleic acid. So deoxyribose is the type of sugar. Ribose is a fancy word for sugar, right? And we know that our DNA is made up of nucleic acids because there are those 4 nucleic acids. What are those nucleotide pairs that make up our DNA? Remember, we have 4 of them. They match up. Yes. A and T and C and G. Those are our nucleic acids that match up in DNA. All right. And the actual rungs of the ladder are the ribose or the sugar. All right. They kind of attach with sugars, and we know sugars are carbohydrates, right? Sugars get a bad rap, but they're also very important. My very non-fancy DNA. Is everybody kind of following me? This give me a green check, red X. DNA is the same in every single cell of our body, right? We have the same DNA in every cell of our body. Every cell in our body does not do the same job. So we talked this week about how there's lots of light switches in DNA, and the process with the operator and the promoter and the repressor, but that we can use DNA in biotechnology. This would be a form of how we can use our cells that were once living to do that. What would happen if we didn't have the same? It would be real weird, Josh, because like If I didn't have the same DNA in my eyeballs or face, like, think about your skin. You could have like all kinds of different tones. Or what if I had like things for a bigger size body, right? I'm a pretty petite person. I'm 5'2". You know what I mean? If I had like other codes for like bigger organs for someone that was, I mean, it could be real funky. Yeah. There wouldn't be I just don't think you honestly, Josh, you probably wouldn't live. Right? Because all of our body organs function together. Yeah. Like there's so much correlation with like why do we breathe? Well, we breathe because we need oxygen. Our brain is in charge of our nervous system, but if our body doesn't give if our respiratory system and our circulatory system don't give our brain blood and oxygen, we die. Right? Like everything in science holds hands. That's why when we teach this stuff, we try to teach it very specifically in order, because we started out the year talking about nucleic acids, right? We started talking about non-living things like nucleic acids, fats, carbohydrates, proteins. And it's crazy because guess what? That was in like August, September. We're in February, almost February. January, and we're still talking about making proteins this week. Because everything in biology works so closely together. Yeah. I mean, honestly, that's why I really love science, is because there are so many little details that we can learn and discover that everything works so closely together. Even like just this gel electrophoresis just is a pure example. Look at how closely that is the DNA of two cells, and when you run it through the machine with that electrical current, it all filters out to look the same. I mean, that's pretty impressive. Biology is detailed and intricate, and you're right. Like if our DNA mutates, right? Like if one of our cells has a mutation and the DNA becomes different, then it gets oftentimes that's when we start, I mean that's a cause of cancer. Does that make sense? Hopefully this is making sense to everybody. Yeah, if we get mutations in our cells, that's what leads to like skin cancer, right? When we don't wear enough protection on our skin, even if we do, the sun can penetrate our skin cells and it hits that fireproof box of our nucleus in the middle of our cells, and sometimes will literally ding one of these nucleotides and knock it out. And that one cell goes through mitosis. Remember, we talked about mitosis. It divides and makes another cell. And now we have two messed up cells. And those two messed up cells make another two cells that are also messed up. Now we have four. And then those four cells, they copy each other. Now all of a sudden we have, see how we exponentially now are copying that mistake. And that mistake, it causes problems, right? It causes disturbances in our body. Now, remember, all mutations don't cause cancer. Some of them can be fixed. Some of them, if they happen once and don't change that amino acid code, they go unnoticed. We all probably have we do. We all have mutations going on. Yeah, Josh, sometimes they can help. Not often, but you can there are mutations that can actually be beneficial, right? We could honestly spend a whole year like all year, like we're studying biology, we could spend a whole year just studying mutations in DNA. And that's honestly how I became a science teacher. I got super interested and I was asking some of the same questions that you are asking. And I went to classes in college and I was like, this is super interesting. I just wanted to learn more, even when it wasn't easy. Like I told you yesterday or the day before, I had to really study. It was hard stuff, but I really enjoyed it because I found it super duper fascinating. All right. Let's go to just like some of the basic steps of fingerprinting. Lizzie, do you want to read these through what happens when we get a fingerprint? I know you had your hand up at one point.
I have a question.
Sure, Jonathan, go for it.
Can these slides also be added into the Gmail, please?
They're already in the classroom.
Not the same ones, I presume.
Yep, nope, they are. They're in there. I'll show you where they're at after class, okay?
But they're not the same, I don't think. Because I don't have that kind of paper in there.
Yep, we'll look for it together, or I can send it to you if that one didn't make it in there for some reason.
Mm-hmm.
All
Anyway,
right.
I do have a doctor's appointment. I don't know what time. I hadn't gotten told about it. What time?
Okay.
It was
Well,
about to be at 12. It was arranged once at 12.
yeah.
I would work with 12, but I'm just telling you forewarned right now. I'm
Okay.
just telling you I don't know if I'm going to be in here.
Well, we don't have class at 12, so that's okay. All right. So let's go ahead and focus back on where we were at. Lizzie or Luna, I know Lizzie had her hand up and I didn't have her read a goal. I was going to have her read this. If Lizzie's not wanting to read, Luna, will you read this? Awesome. Go for it. And Nick, I'll grab you next.
Steps of DNA fingerprinting. The sections of DNA that are cut out are called restriction fragments. This yields thousands of restriction fragments of all different sizes because the base sequences being cut may be far apart, or a long fragment, or close together, or known as short fragment.
Perfect. So remember we talked about this yesterday that we actually have protein enzymes. There are proteins that we use to actually cut the DNA. So if we just wanted to research or study a certain section of DNA, we can actually insert enzymes into a similar tank like those pipettes we saw and put those restrictions enzymes in there, and there are certain enzymes that will cut, and we legitimately call these right here, these nucleotides that don't have a pair yet, they're pulled apart. These are called sticky ends. I love that because it makes sense, right? Because you can almost just like know that you're going to glue those together. So that's how we look at certain sections of DNA. And Nick, you want to read this slide for us about what those restriction enzymes, a little bit more about those restriction enzymes.
Yes, ma'am. Then, the DNA is cut into fragments using restriction enzymes. Each restriction enzyme cuts the DNA at a specific base sequence.
Perfect. Good reading. Thank you. So, what do I want you to know about this? I just want you to know that you can manipulate cutting and gluing together DNA. All right. We can do that for the purposes of study. We can do that for the purpose of biotechnology, like we did yesterday. Remember how we did that yesterday with our plasmid, that circular piece of DNA that we use to grow in bacteria cells. Awesome. All right, we are nearing an end of this. So what I would like to do is get any questions you have about DNA electrophoresis. And then what I'm going to do with the last 10 minutes of class is I would like to take questions from this week. All right, questions about gene expression, questions about biotechnology, genetic engineering, and then there is a gel electrophoresis assignment. All right. So
Is it required?
Yes.
So it's in the 1.07, right?
It is 1.075 is what it is. Okay. So what we're
What
going to
is the length for recording over this assignment?
Yep, let me talk about the assignment and then I can
Okay.
answer the
Okay.
Thanks, bud. Awesome. So if you'll go into the classroom, you should be able to see the 1.07 DNA electrophoresis assignment. Okay. If you click on that, then you can go ahead and start working on that now. There is a document that you just click on, it'll open it up. I believe it's a Word doc. Let me look. Yep, it's
There was something malfunctioning in the 1.075. It's the recording over this assignment.
Okay, so the recording over this assignment is not working because it is not today. We're doing that right now. That's why.
So can you open it?
Well, no, because that won't be available till I post this in there. Does that make sense? This is the recording.
So this we're supposed to do it. Are we supposed to identify what our skin cell stain are?
All right, so pause right here. So I would love for you to stick around. I would love for you to stick around. I will make sure this is a quiet place for us to work. So if we have questions, we can pop into breakout rooms. But what I would like you to do is go in and pull that assignment up right now. Right? That way, if you have any questions while you're working on it, you can. It's going to ask you questions from today's lesson.
What's the assignment called?
1.075 gel electrophoresis.
So is it the PowerPoint?
Nope, it will be a Word document, bud.
It's not in there.
I mean, I am seeing it, so it's in there.
Mhm.
We'll help you find it, Jonathan, okay?
So what is it that we have to do for the assignment in there? Because I have also another assignment that I must do too. That's
Well, you don't have other assignments from me that you have probably not that you have to work on. So since this is our hour for science, we're going to go ahead and work on this right now, okay, bud?
okay.
Okay.
There's some students saying they don't see it.
You aren't seeing it either. It should say 1.075 DNA electrophoresis.
It must have been not gone sent to the other kids neither, I don't think. Because they don't 1.75 1.705 07075.
Gonna make sure my start date isn't my due date. It says due date. Let me make sure.
So what day this what day would they be due?
All right, I'm gonna go ahead and just mute all microphones just for a minute so we don't notes for the recording. I'm going to pop in the classroom as somebody else and see what is going on and why you're not able to see that. Because it looks like from everything on my end that everyone should be able to. You might, because I just opened it, you may have to log out of your OLS and then log back in. So log
There-
out. And
Sorry, there's been a lot of problems with class too. Yesterday, we couldn't get things to load. It kept saying all snap and like nothing was working. So I don't know if it's something like, I don't know. If it doesn't work to refresh, maybe even restart your computers if you haven't done that for a while. But we were having all kinds of problems yesterday too.
okay, well, I'll change the due date on it right now so that we have a little extra time that stretches to tomorrow. But I really wanted you to be able to work on those in class right now, and so that you are able to use the PowerPoint, you're able to use me if you have questions. So keep trying to get in there, and I'm keep trying to do anything on my end that I feel like would keep you from it. The one thing I feel like it's asking is you're going to want to know this also for your thing. It says DNA moves from the blank end to the blank end, right? So you're going to want to know what side the electrodes are on. It's going to be from negative to positive, all right? So it is going to want you to really know that. It's going to want you to know why some move farther. Remember, those are because the large molecules get left behind and the smaller. So molecules that are small move faster than your larger molecules, all right? Yes. Let's answer this one together. Which one of these DNA pieces is the smallest in this sample? A, B, C, or D? Which one is the smallest, and how would we know? Keep in mind that the molecules that are the smallest move the fastest and the furthest. So if it's moving from the negative end to the positive end, we're going to look for the one that is the furthest down the line. Yes, Jenna. Yes, Josh. Yep. So that would be C. All right. So if well number 1 is DNA that was collected at a crime scene, which individual would be the person of interest? Which matches up with well number 1. Which well? See how well 4, these pieces are at the same spot as number 1, 4 and 1. So the person of interest would be number 4. Because 4 and 1 match up. All right, so that will help you too. So if you are not in the assignment, you might want to screenshot this page because this will help you with some of the questions that are on there. All right. Is anyone seeing it yet? Is anyone in the OLS and sees the assignment? Yes. So Rachel sees it. Okay, so make sure not just to refresh, but you might have to go completely out and go back in. It's in your content, Isaac. So like if you go, I would go and completely close out of that browser, or even if you close out but you don't log out. So I go up to the top where your name is, click on that on the far right-hand corner and put log out of the OLS. And then log back in. And you'll see the assignment. It'll look like this. I can screen share. me screen share for you. It'll look like this. Gel electrophoresis assignment. All right. 1.075, you'll click on that. And then it'll pop up a Word document that looks like this. And there you go. See, some of those questions are here. So that's what you're going to want to go over. Almost everything that we did today will be in there.
I'm gonna check on my end too. I don't know. I've been trying to get in. Mine won't even like let me load the classroom, so I don't know what's happening.
Well, I will change the due date to Friday, okay, guys? So I'll change the due date to Friday, and I'll send out an email as well with that Word document attached. Just knowing that, like, it might take a minute for the OLS to, so you'll have to the end of the week to get that done. What I really want you to do is make sure you have answers on this. It won't let you type on it. Try to save it or save as, Rach, and then it will probably let you edit it, if that makes sense. Sometimes it won't let you edit the first. Isaac found it.
And it is working now for me too, so it should be working.
So it will show up. It's there, I promise. I've changed all restrictions of any kind to make sure that, like, even the big bad wolf that comes into our classroom could find it. So it's not you. It's probably you just probably need to log out and give your computer a minute to sync up with all of those things, because I had it hidden before class because I didn't want you to do it without me explaining how a gel electrophoresis works. But it is 9/11 now. I was going to let you early. Sorry for the complications, guys. There are joys of e-schools and there are pains of e-schools. This might be the pain. Sometimes not being able to find things.
True.
So don't stress about it. You have till Friday, right? You have this, so screenshot it. Everybody screenshot this.
It's not labeled as quiz.
Yes, it is not a quiz. It's an actual assignment, like a Word